So this diagram shows you the Western blot analysis. Like I said, it starts with the SDS page, and then the transfer of proteins from the gel and to a membrane. And once that is done, then you will have to block the non-specific binding site on the membrane. After that, your protein will be on the membrane, right? So then you'll screen your protein with an antibody that binds with your target protein. Okay, so we call that a primary antibody. But that's not the end of the story, because your normally primary antibody doesn't have any enzyme conjugated to it or dye conjugated to it. So actually you have to add the secondary antibody that recognizes your primary antibody. Normally the secondary antibody is conjugated with an enzyme. Okay, and then you add the substrate that will give you a signal. So that's how your Western blot analysis works. Okay, and the first step, as this page, we'll skip because we already talked about it. But how about the transfer, how it's done? We'll probably do the electrophoretic transfer. Electroblotting is another word for that. So between the two metal plates, anode and cathode, your gel and the membrane will be placed there. Okay, and then some spacers in between. So it'll be electrically the transfer will be made. So the electrons will carry your proteins from the gel to the membrane. That's how it's done. So it's a more efficient, fast blotting method. So, negative to positive, so the electron will flow from this direction, so gel to the membrane. That's how that'll be done. So membrane, you can use either nitrocellulose, nylon, or polyvinyl lydine fluoride, PVDF. Okay, whichever the membrane you have, anyone is fine, but they have pros and cons, of course. So to transfer setup, of course, you need to use a buffer when you do the electroporating. Normally trace-based, and you have to add glycine. Glycine is the electron carrier, so electrolyte, and then that's a semi-dry buffer. But if it's a total immersion buffer, similar composition, but you have a little less glycine, and you will have a similar methanol, and the trace is the same. Okay. And then once the transfer is done, you need to check if the transfer is done completely. There are a couple of ways of doing it. In the lab, you'll probably use a pre-stained marker, but besides pre-stained marker, you can use something like India ink staining. Actually, your membrane can be stained with India ink, then the protein will show up. That's how you can check the transfer. There's another method called Ponceau acid staining. Ponceau acid is a solution, a dye solution that can also stain your membrane as well. If your protein is there, of course, that means the blotting is done successfully. Okay, so after checking the efficient transfer of your proteins on the membrane, then the next thing you want to do is the blocking of the membrane, meaning non-specific binding sites on your membrane should be blocked. Okay, so how do you block it? In this case, you will need kind of cheap random protein. So normally like low-fat dry milk protein you can use. Casein, there are a bunch of casein in there, or you can use bovine serum albumin if your target protein is not, of course, BSA or milk protein. Of course, you can use these. So yeah, you can use those cheap random proteins to block all those membrane binding sites. Okay. So these solutions may have some residual alkaline phosphatase enzymes, so you should not use these proteins if your detection system is using the enzyme. That's something you need to keep in mind. Okay. So best blocking system is, like I said, 6% milk protein and 1% that solution, and then EDTA, polyvinyl pyrrolidine solution. So anyway, so after heating it 1 hour, if you heat it, I guess the enzyme can be inactivated, then you can use it. Okay, so sodium azide is typically added because sodium azide is an antimicrobial substance. So since you have to incubate it a long time with milk protein and all those nutrient-rich medium, microbial can start growing there. So to prevent that microbial growing, you can add sodium azide a little bit. That's the purpose of adding azide. So once blocking is done, then you will add your primary antibody that recognize your protein. So we call that primary antibody. Normally the dilution will be done 1 to 1,000, 1 to 5,000, depends on how concentrated your antibody stock solution is. And you incubate your antibody with your membrane for about a couple of hours there, okay? And then detection of antibody is normally done this way. So your target protein, and then you have the primary antibody binding to the target protein, right? But the primary antibody doesn't have any color or any signal that it can generate. So normally you have to add secondary antibody that recognize the primary antibody. Okay, so the secondary antibody in this case is normally conjugated with some kind of enzyme that will give you a signal. If the enzyme is given with some kind of substrate that will be converted into a color product or light-based product. Okay, so that's the purpose of secondary antibody. Let's say you add the primary antibody that is made from rabbit. For example, it's the rabbit IgG. If that's the primary antibody, then your secondary antibody may be made from different animal, maybe goat. Okay, but it should recognize a rabbit IgG. So your secondary antibody will bind with rabbit IgG, okay, but it's made from goat. That's what it means. Goat anti-rabbit IgG is your secondary antibody. And that secondary antibody has some kind of conjugated enzyme to it, okay? And then normally those enzymes that are conjugated to the secondary antibody are either HLP, like horseradish peroxidase enzyme. That's a one good enzyme used often. Okay, so because the enzyme can do this conversion, so the substrate can be converted into that product, and that gives the light energy. So the light energy can give you a signal that you can easily detect. And yes, another example, HLP enzyme here can give you the light. So target protein, primary antibody, secondary antibody, and that's conjugated to HLP. And if you feed the substrate, it will give you the light signal. Another enzyme that can be conjugated on your secondary antibody is alkaline phosphatase enzyme, AP enzyme. Again, it can remove a phosphate from different types of substrate. Among those conversions, again, it can give you the light. Okay, so it can do the same thing like HLP, alkaline phosphatase enzyme. Okay. So that's what it is. So, you know, it's antibody-based protein detection method. That's what Western blot analysis is. Okay. So besides Western blot analysis, there are a couple more antibody-based detection method which uses the same principle. So target protein will be recognized by primary antibody, and you can generate a signal if there's a secondary antibody coming in. So that's Western blot analysis. But you can use the similar or same principle in different detection method. We'll talk about those two different detection methods, just like Western blot analysis. First of all, one method is called fluorescent antibody technique. It's called immunofluorescent microscopy. So immuno, when you say something immuno, that means normally antibody. Fluorescent is, this fluorescent molecule, right? So antibody is maybe conjugated with some kind of fluorescent dye. That's what it is. You'll use that. In microscopy, you will use a microscopy method. So basically, it's a in vivo detection method. Okay, so when you make a cell preparation, and you want to locate where the target protein is located inside of a cell, and you can add your primary antibody. Okay, maybe the primary antibody or secondary antibody that recognizes the primary antibody is conjugated with some kind of fluorescence. Then you can identify where your protein is located inside of a cell. That's the protein detection method in vivo. That's what it is, this immunofluorescent microscopy. Okay, so fluorescent tag, okay, that emits the fluorescent signal, okay, that's covalently attached to an antibody. Okay, then you can detect the protein using the antibody, and that detection can give you a signal of fluorescence. That's what it is. Okay, maybe green fluorescence or red fluorescence, depends on what you use. Okay, so So this picture here, so these are the cells that may be going through what, mitosis, right? So green fluorescence you can detect. Okay, so stand for CK, it's a probably some kind of cytogolic proteins, cytokines or something like that. And the DNA, if there's a protein inside of DNA, maybe histone or something, okay, if you have an antibody that recognizes that, and then if that antibody is tagged with the red fluorescent, you can detect them. All the chromosome structures, and this is maybe microtubules or microfilament, okay, that is recognized by the antibody that's conjugated to green fluorescence. So now you can see red and green structures inside of a cell. One is probably the structure protein, okay, and the other one is showing the chromosome structure. Okay, so this is the immunofluorescent microscopy example. Okay. And the second method that's using the Western blot principle is called ELISA. Okay, ELISA stands for ELISA, enzyme linked immunosolvent assay. So enzyme linked, yeah, primary antibody, secondary antibody linked with the enzyme, right? That's what it means. Immunosolvent, immuno again antibody, solvent, solution based assay, just like Western blot analysis. Okay, but instead of using membrane, you do that in the 96-well plate. That's the only difference. Okay, so target antigen, in this case protein, is bound to a solid phage, and the serum containing antibody is added to that, and then unbound things are washed up, and the secondary antibody that recognizes the primary antibody, it normally has the enzyme conjugate to it, and then you give the substrate and you detect the signal, just like Western blot, right? So here you go, antigen on the bottom of the plate, and primary antibody is added, right? And the secondary antibody is added that recognize the primary antibody, and secondary antibody is conjugated. Now you have the substrate that you can detect the signal. Okay, so the binding between the antigen and antibody is detected through just Western blot like analysis. Same thing showing up here. So you can detect the antigen or antibody either way. So in this case, you can detect the antibody presence. Okay, so I can add maybe antigen and then maybe another antibody bind to it. Okay, so it can be done either way. So detection is done on the 96-well plates. So you can see the signal coming in. The signal may be the blue color. No binding will be maybe the white color. So that's how you can see the difference. Okay.